मला वाटतं सुरुवात करूया आपण. 10:30 झालेले आहे. पुढचं शेड्यूल आपलं डिस्टर्ब होईल मग. सगळ्यांना नमस्कार, सुप्रभात. विज्ञान विषयाअंतर्गत विविध घटकांवर आधारित व्याख्यानमालेतील आजचं हे आपलं पहिलं व्याख्यान. यात आपलं सर्वांचं मी मनापासून स्वागत करते. आपल्या पुण्यातीलच आघारकर संशोधन संस्थेतील माननीय डॉक्टर ज्योतिका राजवाडे आज आपल्याला आपल्या पाठ्यपुस्तकातील इंट्रोडक्शन टू मायक्रोबायोलॉजी म्हणजेच सूक्ष्म जीवशास्त्राची ओळख या विषयावर मार्गदर्शन करणार आहेत. डॉक्टर राजवाडे मॅडम यांचा मी आपल्याला अगदी थोडक्यात परिचय करून देते. डॉक्टर राजवाडे मॅडम यांनी पुणे विद्यापीठातून मायक्रोबायोलॉजी या विषयातच BSc, MSc आणि PhD केलेली आहे. इतकंच नव्हे तर त्या मायक्रोबायोलॉजी आणि बायोटेक्नॉलॉजी या विषयात PhD करणाऱ्या विद्यार्थ्यांना मार्गदर्शन करतात. मॅडमच्या 8 विद्यार्थ्यांना डॉक्टरेट मिळालेली आहे आणि 5 विद्यार्थी त्यांच्या मार्गदर्शनाचा सध्या लाभ घेत आहेत. डॉक्टर राजवाडे मॅडम यांचे आतापर्यंत 39 संशोधन अहवाल प्रकाशित झालेले आहेत. अनेक ठिकाणी त्यांची मार्गदर्शनपर व्याख्यानं आयोजित केली जातात आणि याचा फायदा अनेक विद्यार्थी, शिक्षक आणि संशोधक घेत असतात. असा हा मी त्यांचा अगदी थोडक्यात परिचय करून दिलेला आहे, कारण अधिक वेळ न घेता मॅडमचा व्याख्यानातूनच आपल्याला जास्त ओळख होणार आहे, म्हणून त्यांना बोलण्यासाठी जास्त संधी मिळावी म्हणून मी तुमचा जास्त वेळ घेणार नाही. मी डॉक्टर राजवाडे मॅडम यांना विनंती करते की आपण आपल्या व्याख्यानाला सुरुवात करावी आणि मुलींनी जरूर या व्याख्यानाचा लाभ घ्यायचा आहे आणि तुम्हाला जे काय मुद्दे लिहून घ्यावे वाटतात ते लिहून घ्यायचे आणि सगळ्यात शेवटी आपण थोडंसं वेळ प्रश्नोत्तरांना ठेवणार आहोत. मॅडम प्लीज आपण सुरू करू शकताय.
थँक्यू, धन्यवाद सगळ्यांना. आपण फारसा वेळ न घेता प्रेझेंटेशन बघूया आणि मग आपल्याला मॅडम जसं म्हणल्या तसं थोडंसं अधिक प्रश्नोत्तर किंवा या गोष्टी पण बघता येतील. हॅलो, आपण सुरू करूया. तर today I will be discussing few things on introduction to microbiology, which happens to be a textbook chapter. Let's start briefly how to introduce the subject. So let's start briefly about what are microorganisms. Bacteria, algae, fungi and yeasts along with viruses are classified as microorganisms. The word can be split into two words. One is micro and one is organisms. So organisms are the living organisms that we encounter. Of course, viruses themselves are non-living entities, but when a host is available, they will multiply. Another word which makes up the word microorganism is the microscope. And you know that you have seen a light microscope and a compound microscope in your own laboratory in the school. Apart from that, there are several kinds of microscope like dark field microscope, phase contrast microscope, scanning electron microscope, and the transmission electron microscope. I think you all have heard these words in the last few months. Because everybody was at home for some time because of this particular COVID pandemic which is going on. Now, when you talk about microorganisms, the first thing which comes into your mind is infections and diseases. You know that diseases like cholera, dengue, tuberculosis, hepatitis are all caused by these microorganisms. Some are due to fungi, like for example, candidiasis is a yeast infection. Aspergillosis is due to a fungus. Cholera and TB are due to the microorganisms, that is the bacteria. And hepatitis is a disease caused by the virus. Now I have put a star on the COVID-19 SARS-CoV-2. So this is actually at present, we can say that it is a star microbe at present. Why? Because it has actually brought the entire world into its own feet. This is caused by the SARS-CoV-2, which is a virus, which is a virus that you all know. And because of this particular virus, people started knowing about the transmission electron microscope also, because the SARS-CoV-2 was not visible because of its size. It's not visible under the light microscope or the combined microscope. Now, apart from the diseases and the infections, uh, how could you classify microbiology or studies in microbiology? So, when once you have to study the subject of microbiology, it could be broadly divided into what is called as basic microbiology and what is called as applied microbiology. Now, in basic microbiology, we are again dealing with the microorganisms only. But how do they divide? What do they cause? What are the kind of infections? What are the kind of routes of infections? How can you manipulate the microorganism to your benefit? All these aspects are studied in basic microbiology. While in applied microbiology, one studies the applications of all these microorganisms in broad areas. It could be health, it could be agriculture, and it could be the environment. So your actually chapter 7 focuses on the applied aspects of microbiology. Now, there is one particular section wherein you have been asked to recall the process of conversion of milk to yogurt. In common language, or as we all know, as Indians, we were not all familiar with the concept of yogurt. But I know everybody at home is having dahi. So, dahi is another Indianized version of, say, yogurt. That's what you can call it. Now, dahi, you know, all know that आई विरजण लावते कोमट दुधाला आणि त्याचं काही तासानंतर दही होतं. त्यामुळे दुधाची कन्सिस्टन्सी जी असते, सो द मिल्क ॲज सच विच इज अ फ्री फ्लोईंग लिक्विड चेंजेस इनटू दही. You must have seen, forget about the instant ready mixes which are available, idli mix, dhokla mix. But in the classical way, when idli and dhokla were made at home, they were made via fermentation. So the right proportion of the rice, right proportion of the dal are mixed together, are kept rather soaked separately. Then a teaspoon of methi is added in many of these households, and then these dal and the rice are ground in a mixie or in a traditional earthen or a stone pot. So they are ground and then they are mixed. And then in the morning, when you are just about to prepare the idlis, then you find that this whole batter rises up. So this rising up of the batter is nothing but the microbial activity which was initiated in the batter, and the microbes produce a lot of CO2. They produce a lot of acid, and that is why the if you have tasted the batter before fermentation, it doesn't taste acidic at all. But after fermentation, the idlis are becoming soft because of the change in the material consistency which occurred. They are becoming slightly acidic in taste, and they are becoming the dough as such is rising, and that is because of the trapped carbon dioxide. So anything which is fermented, what happens? So there are definitely some microbiological changes which occur. The number of microorganisms increase in the particular product. The number of microbial products also increase. In the whole process of fermentation, there are some harmful microbes which are destroyed, and useful microbes are added. Sometimes in fermentation, flavors are also imparted. So you take examples of idli making or milk to yogurt or dahi, which is there in your described in your textbook. So the milk, when milk is converted into dahi, another important thing what happens is the lactose, which is a component, which is a sugar present in the milk, is converted into lactic acid. And because of this lactic acid, the casein in the milk, because milk is a colloid in form, so that contains lot of proteinaceous material which is in the form of casein. This casein, it's completely mixed when you see a glass of milk, but the moment the lactic acid starts producing, this protein precipitates, and all this activity is due to the lactic acid bacteria which comes from the previous day dahi, or virzan in Marathi. So that this contains lactobacillus, specifically Lactococcus lactis also, which imparts or which changes the lactose to lactic acid. Now there are other organisms also like Streptococcus thermophilus, Lactobacillus delbrueckii, etc., which are specifically used when milk is being converted into yogurt. And this particular yogurt, I have just shown a image of epigamia yogurt, which comes in different fruit flavors. So what is what is done during the fermentation is that when the lactose is converted into lactic acid, some fruit juices are added so that the additional flavor is produced. And the fruit juices also contain some particular material which is characteristic of the food, or a strawberry or a mango or whatever these grapefruits. So all these particular substances help in they also get transformed, and products like diacetyl or acetaldehyde are produced, and therefore the characteristic flavor is you can get the flavor of that particular fruit in the yogurt. Now, there is another process which is cheese making. You all know that just as milk can be converted into dahi, this particular dahi gets could be modified to form what another specifically product. Like for example, the cheese which you all love to eat in the pastas and the pizzas, there is an Indian substitute also to cheese. Indian substitute is paneer. It is somewhat similar to the cheese, but the process of cheese making is little bit different than the process of paneer making. Paneer is referred to as cottage cheese. In your textbook, they have described the process of cheese making. Only thing is, what I have tried to do in my talk here, I have tried to make things a little picturesque, so that we all know that we have a picture memory. So when you grasp images, or when you see images and pictures, you try to store it in your mind or in your brain in a very nice way. So the more pictures that you see, the more your eye gets trained, and similarly your brain gets trained. So to all these microorganisms and their specific names are very important, because instead of Lactobacillus lactis, you cannot get the cheese to be formed using a penicillium culture right in the beginning. So the microorganisms, their names, their activities are very important, and the making of pictures and pictorial representation often helps us in remembering the things very quickly. So briefly, if you want to, how is cheese making, or how is cheese made? So first milk is there, then they add the culture of Lactobacillus lactis or Lactobacillus cremoris and Streptococcus thermophilus. What happens with these cultures is that souring of milk occurs, and then the milk solids precipitate. Then there is another process. Like here in dahi, once the milk is formed and we just try to eat it at the same time. Once it is formed into dahi, we try to consume it. But here in cheese, this particular protein, which is forming a very tight mesh, has to be little bit released, partially released. So this activity is done by a enzyme, which is a protease enzyme from the fungal species or the fungal cultures. And then what is happening after the net formation? a different type of curd is formed. Then the curd is pressed, then it is cut, and then it is either scalded or cooked, and then you can get a like a well formed tough curd, and then the whey is separated. Here another picture I have shown, the same thing is there, and then once this the setting of curd occurs, the whey and the protein, you can refer to the right diagram on the right, and then we have the whey and the curd mixture. The whey is drained, and then what happens is some amount of salt is added to the curd. So this particular salting occurs, then everything, the whey is drained, and then mold is, all this mixture is now put in the mold, and then it is stored. So storage is a very important aspect of cheese making, and this is process is called ripening in cheese. After the ripening, the cheese is packed and spread and then marketed. Now, sometimes in pictures you might have seen that some cheese looks blue in color. Now, why this cheese looks blue in color? Because there is a specific microorganism called Penicillium roqueforti, and this microorganism is added, and because of that, the cheese looks blue. We have seen the Tom and Jerry, that comic serial or the comic strip on video, and we have read it in books also. In that, always they show that the cheese is having holes in that, and that hole, the cheese with the holes, is nothing but camembert cheese. So there are different kinds of cheese, and the different kind of microorganisms. This is the fungus, which is another fungus, Penicillium camemberti, which is added to produce camembert cheese. Now, there is another question in your textbook which says that just enlist the different types of cheese which are available in India. So, in India specifically, freshly prepared cheese are always available. For example, the cottage cheese is available, cream cheese is available, mozzarella cheese is available. And I have shown you different types of cheese in this particular picture. So after ripening, we have discussed the process of ripening in the earlier slide. So if the ripening is for 3 to 12 months, then we get a semi-hard or cheddar cheese. And if the ripening is for 12 to 18 months, what we get is a hard Parmesan cheese, which is there on your pizzas before they are baked. Now, in a nutshell, I would like to show what happens when milk is first converted into curd. What happens is, like lactose hydrolysis occurs, there is some amount of lipolysis also, because the milk which is used for the curd formation contains some lipids. So these lipids are again broken down by the microorganisms or converted by the microorganisms into glycerol and free fatty acids. There are a lot of mineral changes which occur. Like, for example, the mineral level like calcium, potassium, magnesium, phosphorus, zinc all increase if we eat more of dahi rather than milk. So you must have sometimes heard in your mother or the elders in your home saying that तू दूध तर पीत नाहीयेस, निदान दही तरी खा, दही तरी छान आहे तुझ्यासाठी. Why? Because the mineral content is increased. Because of these microorganisms, the enzymes are increased, and that is why dahi has a more positive effect on the system as compared to milk. But I think, in my opinion, we should always eat dahi for its probiotic activities, and also if you like milk, you can have milk also. Specifically, when the milk is made into dahi, several antibacterial activities are also associated with this dahi. Then there are a lot of vitamin changes also occur. All these microorganisms from the lactobacillus category, they produce vitamin B, and vitamin B is essential for a very essential vitamin, and therefore we should consume lot of dahi. Then the first thing which occurs, which we already discussed, is proteolysis, or degradation of the precipitation and degradation of the protein. So milk proteolysis occurs where proteins are converted further into peptides, amino acids, and peptones, which are very easy to digest. Sometimes you must have heard this particular comment also. त्याला दूध बाधतं, त्याला दूध आवडत नाही त्या व्यक्तीला, किंवा he is lactose intolerant. So there are some individuals who cannot tolerate lactose or who cannot metabolize lactose. Therefore, then they get the situation like diarrhea, then they get rashes on their skin. So in those conditions, lactose or milk is to be avoided. And for these individuals, dahi is well tolerated because lactose doesn't remain as lactose in milk, it is converted into lactic acid. Now, there is another question in your textbook which says that you try to find out some information on the various kind of cheese which are available and their specific uses. So, for your information, I will be like sharing this particular presentation with madam also, and then this is some compiled information which will help you to remember things well. For example, the mozzarella cheese which you eat has a very mild flavor. It is soft and it is chewy, and therefore it is used in fresh salad vegetables, and in dry form also it is used as topping on the pizza and other baked dishes. Like another example, I would not read the whole table out for you, but another example I can give you is the feta cheese. The feta cheese is particularly a Greek cheese which is bit salty. The texture is very soft and creamy, and it is used in grilled or grilled salads, or sometimes raw cheese also is used in salad or marinade with olive oil and several herbs. So few things, the diet, the combinations are fixed. Another thing at the last thing I have put is the cottage cheese or the paneer. So the paneer, which we all relish as paneer paratha and all the various dishes, paneer butter masala. The paneer has a low fat. It is slightly salty. So the persons who are on diet are asked to eat paneer and not milk. So, because it has low fat and high amount of protein, cottage cheese or paneer is a popular Indian food. Along with this particular cottage cheese and paneer, Kalari and Kalimpong are also very famous. These are another kind of cheese which are the Kalari is from Jammu and Kashmir, and Kalimpong is from the eastern states, that is the Meghalaya and the eastern states of India. Here Kalimpong is very famous. So these are the typical Indian cheese also, and they are also liked by everybody. Then we at times eat the cheesecake or tiramisu. So this is particularly a Italian cheese which is called as mascarpone, and that is used to make this particular cheesecake. Then there is a very one small question which I actually I was amazed reading your textbook also. So different things, because after doing microbiology also I have never referred to now textbooks. This is the first time I was reading standard 10th chapter. My I have a daughter, and she completed her 10th long time ago, so around 10 years ago from now. So after her schooling, I have not seen the my inclusion of microbiology in so detail in your textbook, but it is very good, I found. And I thought that I should be giving some more information to you also, because some questions are there, like they have asked or discussed the importance of mutant strains in the dairy industry or in general microbiology. So why these particular mutant strains are there? So mutants are, you can say that you have heard about mutants now in the COVID-19 pandemic also. So alpha, delta, beta, all these are nothing but the mutants. So there is some change in the DNA sequence or the nucleotide sequences which has changed the structure of the microorganism. So these particular mutations occur naturally also, or sometimes they are man-made. So there are certain chemicals which can be used to induce mutations, and these mutant strains are used in dairy microbiology, for example, because like these mutant strains are made resistant to viruses. So just as viruses are attacking the human beings, like SARS-CoV-2 is the example, the viruses are also attacking bacteria. And the viruses that attack bacteria are specifically called as bacteriophage. So sometimes due to some small, some what do you say, some lapses or some kind of improper sterilization, sometimes the phages could be introduced into the milk, and then the whole process will go bad. So then if you are adding the microorganisms from outside, why not make them resistant to viruses and add? So this was the concept, and therefore the introduction of mutant strains is more important. So some mutants have very high tolerance to acid or increased temperature. In certain cases, the wasteful pathways, because the microorganisms also spends energy. It has to be given glucose or some other products to eat, otherwise it cannot divide. So some mechanisms are wasteful, and you don't want those mechanisms to be done. So certain pathways can be inhibited or deleted in mutants, and the pathways which are favorable to us can be more active in some cases. So therefore, the mutant is very important. And the last point, that proprietary products can be produced. So what you cannot actually, whatever you isolate from the natural environment, it is not patentable. Because it is a biological form, it is not patentable. But now the scientist has said that yes, the culture which I have produces lot of penicillin, which is more than any of these reported strains. So the penicillin that is produced by this culture is a proprietary product. So proprietary product can be produced using mutants. So in this particular slide, I have also added some of the, because we are into dairy industry and application of microbiology, I have also enlisted some advantages of the fermented foods. So why we should use the fermented foods? The main reason is that fermentation increases the shelf life. Like for example, the milk, if you keep for a longer time, it will get very bad, because more unwanted bacteria will come, and then they will turn the milk into curd, but that curd will not taste nice. But if you convert milk into curd, then the curd can be consumed for 2 to 3 days also. You can keep the curd in the refrigerator and eat after 3 to 4 days. Nothing will happen to that curd. So properties of the original food change after fermentation, and sometimes many of these enhancement in the organoleptic properties, etc., can be obtained. Then we need to focus on one more aspect when we are considering all this dairy microbiology. We are going to there is a topic of probiotics which is there in your textbook. Now we in the past few slides, we already talked about the lactobacillus. So apart from lactobacillus, there are other cultures which are called as lactobacillus acidophilus, then lactobacillus casei, bifidobacterium, bifidum, etc., which are known as the probiotic cultures. So what do these probiotics do? They maintain the intestinal bacteria. That we all know that if we have a good intestinal microflora, or if our digestive system is good, then we don't have any problems. That is the first thing which is said by all the doctors also. So you have to maintain a so you if you don't want to suffer from any infection or something, you keep your digestive system proper. That is what they said. So proper means what? So because the intestine already contains thousands of microorganisms. Now all these thousands of microorganisms are contributing in their own way to our well-being. So if we have these microorganisms, what happens is that they help digestion, They decrease the harmful microbes. Sometimes Clostridial infections are there. These microorganisms or their products will kill the Clostridium. Then they improve the resistance to immunity, resistance to infections. So these microorganisms increase our immunity. They lower the ill effects of the harmful substances formed during metabolic activities. So whenever our metabolism is occurring, then certain products which are harmful to our body are being produced. So these bacteria destroy the harmful products also. So one should consume the probiotic products. So what are the probiotic products? Yogurt, kefir, dark chocolate, pickles, oils, corn syrup, artificial sweeteners also contain probiotics. Now, one of our batchmate has even started a company which produces chocolates filled with the probiotic. So once you eat the chocolate, you already have good bacteria going in your stomach and helping you. So you all know that several medications also go with a probiotic. So sometimes if person visits the doctor and for treatment of some infection, and the doctor is prescribing antibiotic, then along with the antibiotic, the doctor is also prescribing a probiotic. So what happens during antibiotics is that the resident or the original microflora which is present in the intestine gets washed away in the during the antibiotic treatment. So this probiotic will try to re-establish the flora. असं तुम्ही ऐकलं असेल की अँटीबायोटिक घेतलं आणि पोट बिघडलं म्हणजे काय झालं, ते बॅक्टेरिया जे पहिले होते ते सगळे निघून गेलेत तुमचे, आणि दॅट इज वाय यू आर हॅव्हिंग डिफिकल्टी इन डायजेशन ऑफ फूड. So it after antibiotic treatment, always the probiotics are always prescribed. Also in nowadays poultry feed are also coming, which have the probiotics. Supplemented poultry feeds are there in the market. So they are fed to the chickens or the hens, and we get the meat. So this is a slide which is showing the health benefits of probiotics. It stops diarrhea. It is good for the heart and the mental health. Then it regains the immune system. Then the allergies and all these things, these problems are also avoided. It also helps weight loss and promotes the activity of good bacteria in the intestine. So these are some of the effects of probiotics, and one should always consume probiotics. The best form and the easiest form available is dahi at home. So this is one of the slide which is actually showing what these bacteria do. This is our intestine, the one in the yellow color. These are the cells, and this is the lining of the intestine, and in this lining there are lot of bacteria which are present. Now with the addition of probiotic, you can see that this intestinal lining we are increasing by adding these probiotics, and these probiotics help to kill these harmful organisms. They produce lot of bacteriocins which will destroy the harmful organisms. Then they promote the immune cells, that is the dendritic cells, the T cells, then the cytokines which are present in our own body, and this is how the probiotics help. Now we have already discussed, so this slide I'm going to skip very quickly, because what happens during fermentation, this I have enlisted here. There are some activities which are enhanced, some activity which are decreased, and some new activities which are formed. So this is the nutritional enhancement after fermentation. Now till this time we have discussed the fermentation mainly of milk, but meat also is fermented in so what are the changes when the meat is fermented? So this is shown in the form of this particular diagram. The lipolysis, proteolysis is just similar to the what happens in milk, but additionally in fermentation of meat there is color stabilization, decomposition of peroxides, flavor and aroma development, and preservation. So this is the main thing which happens. Nitrates and nitrites are also reduced. These nitrites are harmful to the body, our body. So reduction in nitrates and nitrites increases the shelf life of meat also. Then another product of fermentation is bread. So we all know that bread looks like this, and bread has a rise. All these is because the bread is fluffy because of the pores, and these pores are because the carbohydrate present in the grain flour is converted into carbon dioxide. So in the process of bread making, this culture of Saccharomyces cerevisiae, that is the yeast culture, is very important. It is mixed with salt and water, warm water usually. Some amount of salt is added, and this particular culture is made active. Some small amount of glucose or sugar is also added at this stage many times. So this particular Saccharomyces cerevisiae is very much important, and this increases the nutritive value because Saccharomyces has lot of vitamins, enzymes, and micronutrients which are present and which are important for our life. So this is the starting culture. It is mixed with the grain flour and allowed to stay for a while. So the activity of Saccharomyces starts, and in the activity what happens is carbohydrates are converted into carbon dioxide, and this rise is there, and then finally the material is baked to make the bread. Similarly, other products which are obtained by fermentation are vinegar, soy sauce, and that we will discuss in the next slides. So here in this slide, we have shown the production of vinegar by fermentation. Now what happens is here two types of bacteria are used. First is the gluconobacter and the second is the acetobacter. So what happens is for vinegar fermentation, we can use several fruits. Sometimes the fruits are not consumed, they are produced in excess quantity. So this particular, like for example banana, we can produce lot of banana, there is lot of surplus banana which is available. Then the first step is the alcoholic fermentation, where the yeast or the Saccharomyces cerevisiae is used. So here what happens is in the complex sugar from the banana is getting converted into ethanol due to the activity of Saccharomyces cerevisiae, and then this is followed by the acetic acid fermentation, which is there because of the acetobacter. So this particular acetobacter will form the acetic acid during fermentation. So the final products which will be obtained are acetic acid. At some time, ethanol could be produced as a byproduct of fermentation. Also, you could have carbon dioxide which is produced in vinegar fermentation also. Now, if you replace these bananas by apples, then what you get is called apple cider or apple cider vinegar. So vinegar from apples is called apple cider. Vinegar from apples is called cider, and it is the prefix apple is given to it. So what is vinegar? Vinegar is nothing but 4% acetic acid. And in many of the pickles you might have seen that the vinegar is added, and that is just because it increases the sourness of the pickle. It reduces the it helps in reducing the oil content of the pickle, but makes it last for a longer time. So these are the advantages of using vinegar in pickles, and this vinegar is produced using microorganisms. Similarly, we can have the various fruit juices like cane molasses, maple juice, grape juice, then starch roots, all can be fermented by using Saccharomyces cerevisiae to form ethanol. Another interesting example is that soy sauce. Soy sauce is brown in color, and we all have tasted soy sauce and various Chinese products made from containing this soy sauce. The soup also contains some amount of soy sauce. So how is it made? So there is this flour of wheat or rice which is taken, and it is mixed with soybean, the flour of soybean, and both this is fermented using Aspergillus oryzae. So this particular organism or the fungus is used to make soy sauce, and after fermentation of these various flours of wheat and soybean, what you get is the fermented product, that is the soy sauce. Then apart from all these, microorganisms can also be used in preparation of various beverages. Beverages means tea, coffee, vinegar, wine, cocoa. All these are nothing but beverages. That is the soft drinks which you all consume. So what is the role? Like for example, in the Lactobacillus brevis, what happens is the coffee seed has it's a fruit and containing the seed, and these seed are there in this pulp. So the pulp has to be treated using the Lactobacillus culture, and then the seeds are easily separated from the fruit of coffee. And then after the seeds are separated, seeds are roasted and ground, and then you have coffee. So whatever the coffee you are drinking, you must remember that some portion of the coffee was made by our friend, which is Lactobacillus brevis. So definitely, during my lecture, always I tell that there is a full scope for all of this. Whatever you study in the textbook, you can get different jobs. You can be starting your own companies also, if you have information or if you have knowledge about what the good activities that a microorganism can do. So this was the aspect of food microbiology, and this was not only in the preparation of food, but food microbiology also involves laboratory testing of food, the microbiological analysis, and finally the quality of food. So whatever you are eating has to be a quality product, and all this cannot be achieved without the microbiology person. So there is lot of importance, lot of scope for microbiology in daily lives also, and earning your bread, because you are 10th class students, you are going to go for higher studies, and then you can start. So you can have different avenues you have to explore and find out. This is the basic information which you are gathering in your school days. All this information has to be used in your life, which is to follow young age and the old age. And here you have to follow, you have to be the earners, bread earners for your family. You have to support your family, and therefore you need to be either employees or employers somewhere. You have to work, and this discipline or this learning can help you in getting jobs also. So you can choose your career by looking at different things and then decide what best suits you. Another aspect which is there in your textbook is that the various microbial enzymes are discussed. So the all the names are given in your textbook itself. I have just put them in the form of a picture. And why these microbial enzymes are important? Because they have very specific action. Then they are wide at various pH. Some enzymes can work at pH 2, and some enzymes can work at pH 10 also. So the pH, temperature, pressure, all these are the various abiotic factors, or these uh, microorganisms can or their enzymes can be definitely used at a different set of temperature, pH, and we can have various products made from using microorganisms. Here, this is a table from your textbook only. Only thing I want to point out is this contains the word amino acid. Actually, lactic acid, itaconic acid, gluconic acid are not amino acids. They are organic acids. So sometimes glutamic acid, to an extent, I can say that it is amino acid, but all the rest are not. And therefore, this particular table has to go with this correction. Now, all these kind of acids uh, are used in our daily life, and they are produced using microorganisms. For example, gluconic acid. uh, Sometimes person needs calcium or iron supplement, and if you read the medicine, there it is written that calcium gluconate, iron gluconate is written on the medicine. So what happens is this gluconic acid is of microbial origin that is mixed with calcium, and then it is converted into a medicine which can be given for the patient. So in this way, many of these products are useful. Citric acid, for example, when you want to make instant dhokla, you use citric acid in that particular batter. So citric acid gives the acidity, and it helps the production of carbon dioxide also. So the dhokla gets a good rise, and citric acid imparts the slightly sour flavor to the dhokla. So where does this citric acid come from? It comes from Aspergillus niger. What is the food for the Aspergillus niger? The sugar from the molasses and some amount of salt will convert. So this our friend Aspergillus niger will convert the sugar molasses to form citric acid, and this citric acid can be used in the toffees and the chocolate and our dhokla also, instant dhokla. So this is how we should learn and things and keep things in mind. Try to remember and then write the question papers or the answer the questions in the examination. Then this is a very colorful slide. I will show you this are the different types of microorganisms are there in this particular petri dish, and this petri dish contains all these different colors. So these are nothing but the pigments. So many of these fungi are known to produce pigments, and these pigments can be extracted. They can be produced by fermentation, and then there are processes like solvent extraction, concentration, and characterization, purification. Everything is done, and then these fungal pigments can be used to color the ice creams and your fruit juices. They can be used in healthcare because your tablets are also colored at many times. Cosmetic applications are also there for the pigments, and lastly they can be used to color the clothes also. So microbial pigments are a very interesting area, and many of these microorganisms produce different colors. And similarly, one more product of microbial origin are polysaccharides. So the word poly is many and saccharide is sugar. So many units of sugar come together and form polysaccharides. And microbial, because these polysaccharides are of the origin, they are produced by microorganisms, we are calling them as microbial polysaccharides. So xanthan, which is a gum, which is a best example of a microbial polysaccharide. And this particular polysaccharide, like this is the culture Xanthomonas, which ferments starch or molasses and converts and forms xanthan. So this is a colony of the culture. You can see that how shining it is. This is how the culture looks in a plate. And then finally, when the xanthan is isolated and purified, it looks like a white powder, which is water soluble and has very high viscosity. And because of this high viscosity, it is used as a thickening agent in ice cream, pudding, chocolate, milkshakes, and all, even instant soups. So the ingredient list will have this particular polysaccharide mentioned as the food product ingredient. Apart from the thickening agent, it can be used as a pigment, it can be used in production of paper, it is a weedicide, so it can be used in agriculture also. It can be used in fertilizer as a slow release compound. It is even added to toothpaste. So one compound and several uses. This is the beauty of the xanthan, which is produced by the microorganisms. You might have noticed that in some cases I have underlined the words, and in some cases I have put italics, or I have written the words in a different form. So why have I done that? So while writing the paper, when you are you cannot write in a curved form in between. So it is best to write the microorganism's name and put a dash below it. So that dash will tell the person who is checking that yes, this is a microorganism name, and the name of the microorganism has to be put either in the italics or underline. So underline is easy for us to do. So you should underline the name of the microorganism. So this is the slide showing different fungi, yeast and bacteria. All these are microorganisms, and they are producing lot of compounds, and these compounds are very important on a industrial level. So this particular slide again is from your textbook. Just to like link it up, I have used the earlier slide. It shows different substances which can be obtained using microorganisms. We have colors, emulsifiers, various essence. So we all enjoy vanilla ice cream. So vanilla ice cream contains vanillin as the essence, and this essence is nothing but microorganisms. Actually, the source of vanillin is the plant, but we cannot grow so many plant and have the vanillin, and therefore the scientists have come up with the process to manufacture vanillin. The color, the flavor is there, but the it is obtained using the microorganisms. So this is how microorganisms can be used. Even biodegradable plastics can be made using microorganism, and this is poly beta hydroxybutyrate, which is an important compound which is made by the bacteria and used in the biodegradable plastic. Similarly, we have various antibiotics which are produced by microorganisms, and this is a plate which will show that the penicillium produces penicillin, and that inhibits the growth of the staphylococcus. So this was the observation by Sir Alexander Fleming in 1920, and this is how the antibiotic were discovered. So otherwise this is the growth of the organism, but because of the penicillin, the production, the staphylococcus doesn't grow near the culture. Similarly, there are various aspects like biogas production. This is also a microbial activity. Here there are different sets of microorganisms. The first phase is the acetogenic phase, where microorganisms convert the complex material into volatile fatty acids, and acetic acid and other volatile acetic acids, volatile organic acids. And then comes the methanogenic phase, where actual from these acids, methane, carbon dioxide and hydrogen is generated, and the leftover of this, so the gas which is formed, is used for cooking, for example, or for converting into electricity or heat. But there is a lot of organic fertilizer in the form of slurry which is being formed after biogas production. So it is the excrement from the livestock or the food processing company, even the garbage, everything can be converted into biogas. Now this is a picture which shows the importance of microorganisms in the sustainable energy. So microbes are a very important for the production of various fuels, and use of microbially produced chemicals in plastics and even food products is also there, but some kind of materials are also used as fuels. And we have liquid fuels in the form of ethanol, butanol, which are produced by microorganisms. Methane, hydrogen is produced again by microorganisms. And there are other chemicals like acetone, alcohol, fatty acids are produced by microorganisms, and they can be used as fuel substitutes. So what the importance of biofuel is that it is a renewable source of good energy, because you see that this is a closed figure, nothing is going really out to the environment. So even if there is a liquid fuel which is producing the CO2, this CO2 is going to be consumed by the plants which are going to be there on this earth. So the plants have to be there. They are the carbon dioxide fixers. They can harvest light energy, and then the whole process of obtaining fuels can start. So there are three kinds of fuels, and these are easily available and plenty of quality. And these are the reliable fuels for the future. So you should plant more trees, have the microbial activity, and manufacture your own fuel. So the stocks of petrol are very much dwindling and the prices are increasing. So to combat this, we can use microbially derived fuels. Then there is a very important component of waste degradation using microorganisms. So these are the typical questions which are asked in your textbook. So why we should segregate the waste, wet waste and the dry waste at each home? So the thing should start from yourself. Why do you depend on the municipality to separate the waste? You can separate the wet waste from the dry waste. So segregation will give good, faster treatment. So that is why you have to segregate the waste. Another thing is what is done with the segregated waste is the question. So if the waste contains glass, it can be recycled. If it contains plastic, it can be converted into some fuel, or even clothes again, from the plastic material, or the PET bottles can be made. So there are different methods. Like for example, there is a bad camera or bad mobile phone which you want to discard, but you want to discard, you discard it at the appropriate place, because the mobile phone which you are using contains lot of precious metals. These metals can be recovered from the waste, and therefore it is important to segregate any kind of waste to have effective waste management. Now even our Pune Municipal Corporation is going to have a drive that if you segregate the waste and put it into the proper containers, then you will earn money. So one bottle you put in the glass container, in the area designated for the glass, and you will get 1 rupee. So waste also can be converted into money, and nothing is garbage. It is important to segregate and manage the waste. Now we can use microorganisms to reduce the pollution. And this is a typical diagram of a landfill site, which is there in your textbook itself. And what can be done using microorganisms is, microorganisms will have two types of digestion can occur in the sewage treatment process. One is the activated sludge and another is the anaerobic digestion. So you can have reduction in the pathogens, harmful bacteria will be killed, and products also can be degraded. We can have bioremediation of sites also, because you must have read in the newspapers that Urali Devachi is a site which is getting completely dumped with the waste, and all these kind of landfill is there, but that is also accumulating in large numbers. So what another alternative is to segregate the waste, prepare biogas in the premises itself. In our home also we can make compost, and we can degrade all the bio waste which is generated in our own kitchens. So decreasing the volume of garbage is important. You can decrease the volume of garbage even at home by preparing the compost. Similarly, in large areas we need advantages, because uh, of compaction, large materials can be transported from one place to another, and then it is easy for the microbial degradation also. So this kind of questions are there in your textbook, and some clues or answers I have tried to give here. Another thing is the use of microbes in pollution control. So there are a lot of oil spills in the sea, and all because of burst of oil tankers or other problems are there. And what happens is, because of all these life spills, the marine life is affected. See the particular bird, all its feathers are covered with the oil. See the black color tar which is there. And here, in such situations, our microbial friends can come to help. Like for example, Pseudomonas species, Alcanovorax orcumensis, they can degrade the hydrocarbons. They can form CO2, that is carbon dioxide, which is escaping into the air. Water, which is required. Also, pyridine-like compounds are degraded by these microorganisms, which are our friends, and they can help in managing all these spills. Even the PET bottles also can be degraded by these bacteria. But then you say that PET is not good. Then, but there are organisms. Why can't we use them? But you will have to consider that the process of production of carbon dioxide during fermentation of bread is only 4 hours. But here, for degradation of these polyethylene terephthalate, The microorganisms will degrade it, but they will take hundred of years. So although the microorganisms are there, the processes are very slow, and therefore efforts are being made to uh, hasten the process of degradation. But at present, this is the situation. So it's best not to pollute the environments and try to use biodegradable things that can be composed even at decomposed even at your own home. Another important aspect which has just to be touched, because this is little bit complicated, and this is called bioleaching. It is the extraction of valuable metals from low grade ores. So our lab initially, about say 20 years ago or so, was engaged in all these research on bioleaching, and here the microorganisms can use the sulfur in the low grade ore, converted it into sulfate, and during formation of this sulfate, what is happening is the pH is reducing. It is like the microorganism are producing sulfuric acid, and because of this sulfuric acid production, the copper in the ore is getting leached, and you can see here beautiful green blue areas, and these blue areas are nothing but copper sulfate or more solution, which is due to the microbial degradation or leaching of activity. So here is another concept where microorganisms, they are not visible to our eye, but they are our farmers friends. So you must have seen that whenever we get methi from the market, the roots of methi are having these kind of small globules. So these globules are nothing but called nodules, nodules. So these nodules contain these bacteria which are called as rhizobium, and these rhizobium give the what they do is they do a very important activity. They fix the nitrogen. That is, nitrogen is present in our atmosphere. We know that 70% is nitrogen, and this nitrogen is converted into plant assimilable compound, that is ammonia. Ammoniacal nitrogen, NH4, is produced, and these microorganisms in turn get products of photosynthesis for their own living. So this is a symbiotic association. I have shown here a plant of pea, that is matar. This is also another leguminous plant which like methi only, which here you can see the presence of microorganisms or nodules containing the bacteria. And because of all this bacterial activity, what happens is no chemical or less chemical fertilizer is required. There is no pollution, and that is why these microbial fertilizers are to be used on a large scale. Now this culture and this plant, they are, like for example, forming a close association. They are friends with each other. They are called as symbiotic bacteria, and some bacteria are asymbiotic. So azotobacter is one such organism which can is a free living organism also, and but it converts, it can use atmospheric nitrogen. So there is symbiotic nitrogen fixation as well as asymbiotic nitrogen fixation as part of biological nitrogen fixation. Then these microorganisms are a part of all the nutrient cycles in nature, and they help in the biogeochemical cycling of the materials, and therefore they are very much important. They help in destruction of pesticides also. Therefore microorganisms can be used as seed dressing material. They can be used to supply nutrients to the plants, etc. Similarly, one more aspect is there, that microorganism can be used to control the pest. So we know that when we bring हरभरा at home, we find that there is some अळी, हरभऱ्यामध्ये अळी असते, किंवा मटारमध्ये अळी असते, किंवा तुमच्या झेंडूच्या फुलात अळी असते. त्या अळ्या काय करतात, तर झाडांची पानं खाऊन टाकतात, आलेलं पीक खाऊन टाकतात. त्यामुळे these larvae are called the pest, and these pests can be controlled using the chemicals. But chemicals are poisonous, chemicals are toxic, and sometimes microorganisms also or their products can be used to control. So one of the microorganisms which is very much helpful in this is Bacillus thuringiensis or BT. You all know BT cotton. So what is BT? So BT is nothing but a short form of bacterial thuringiensis. This is a kind of protein which this organism is producing. This organism produces this diamond shaped protein. And what happens is when this organism is put on the leaf, for example, so this caterpillar comes and feeds on the leaves. And in the caterpillar, the enzyme and the protein undergoes solubilization, activation, and finally, in the last part of the caterpillar gut, this there is pore formation, there is leakage, and all the finally it result into the death of the insect. So this kind of biological control of pest is possible using microorganisms like Bacillus thuringiensis. Apart from these organisms, there are certain fungi, verticillium for example, metarhizium, they can be used for control of these insects. Even some viruses can be used. For example, nuclear polyhedrosis virus that can be used as to control the butterflies and the moths, which are dangerous, which are causing diseases to the plants. So another important point which I felt in your questions answers was that they have given this kind of diagram. This is a conceptual picture in environmental management, like how the microorganisms can be used in environmental management. So these are the various aspects in which microorganisms can be successfully used in the environment. It could be in the form of for heavy metal recovery, biodegradable plastic, handling the oil spills, etc. So this was, I thought that I should cover this chapter. Actually, this chapter is quite interesting. It's quite big, and I tried to make it as picturesque as possible. For I hope you understood today's talk. So if you have any questions, I can take the questions now.
मुलींनो तुम्हाला काही शंका असतील तर ज्योतिका मॅडमला विचारा तुम्ही.
I think I'll stop presenting now.
Yes, ma'am, that will do. Yeah, I think we should over this lecture now because we are supposed to admit other students of other classes. We are going to start their lecture at 11:45 and it's already 11:37.
Yeah.
So I think we should stop for right now and we should start with the, actually you also might have need some break for 5 to 10 minutes because you have talked so much and I really felt so nice that you gave such a nice information to all our students. It's really very deep and the topic is really very vast, but you covered it very nicely. You touched all the topics, like right from the how microorganisms are useful in food industry, how they are useful in pollution, to control the pollution, and how they are useful in agriculture. And of course, it has very nice career opportunities in future. And so our students, they might have been come to know about that also. So I'm really very thankful to you for giving such a nice lecture to our students. And we are eagerly waiting that very soon we will invite you for next lecture also.
Yeah.
So students, any query, any question, so that you can ask ma'am. She will answer. Okay, I don't think so they have any question or query, but if they tell us any question or query, I will let you know.
They will, definitely. Yes, madam.
Yeah. Okay.
So I will also leave the call and rejoin.
Yeah, no problem. No problem. Yeah. Thank you, ma'am. Thank you very much.
Thank you.
Yes. मुलींना तुम्ही लेफ्ट झालात तरी चालेल, काही प्रॉब्लेम नाहीये. आता दुसऱ्या वर्गाच्या मुली जॉईन होणार आहेत आता.